Friends, welcome back to my channel, and today in this video we are going to study about linkage versus independent assortment, and in this we'll learn about linked genes, crossing over with linked genes, and recombination frequency. So first of all, what are linked genes? Linked genes means these are genes that are so tightly linked with each other that they do not obey Mendel's law of independent assortment. What is Mendel's law of independent assortment? It means that genes assort independently. For example, if we have one gene for height and one gene for color, we have two alleles, capital T small t, capital C small c, for example. So, the alleles of one gene can independently assort with the alleles of another gene, and we get the ratio 9 is to 3 is to 3 is to 1, where 9 is when both the characters dominant, 1 means both the characters recessive, that means parental characters. These two are parental characters. What about the middle 3 and 3? These are hybrids. That means one character dominant and one character recessive. For example, in case of first three, if T is dominant, then C will be in recessive state. In the next three, the C will be in dominant and T will be in recessive state. So 9 is when both the characters are dominant. The next three, when one character is dominant. The next three, the other character is dominant. 1 when both the characters are recessive. So this 9 and 1, these are parental phenotypes. But this 3 and 3, these are hybrids, combinations. One character recessive, one character dominant. Linked genes are the genes that are tightly linked. So suppose if these two are linked genes, in that case, these two genes will be so tightly placed that they will not show Mendel's law of independent assortment. That means we won't get this 3 is to 3 middle one ratio because they won't recombine. Recombination can only happen when the genes are not linked. If the genes are linked, they cannot recombine because they are already tightly linked. So this middle 3 is to 3 ratio we won't get in case of linked genes. So linked genes are the genes that do not obey Mendel's law of independent assortment. These genes are inherited together. That means though they are two separate genes, but they will behave as a single gene. So this will considered as one gene. So within one gene, recombination cannot happen. Recombination can only happen between two genes. One gene is present here, the second gene is present here. So recombination can only happen between two genes. Within one, the recombination cannot happen. But if the two is present inside this circle, for example, these are too close or too tightly packed with each other, then recombination cannot happen. These genes are located close together in the same chromosomes. They are called linked genes, and linked genes belong to same linkage groups. Now, linked genes do not assort independently. What we know about law of segregation and independent assortment? Law of segregation means allele separation during gamete formation. Independent assortment means alleles of one locus assort independently from the alleles of other locus, which we learned just now. And also in our earlier videos on law of segregation and independent assortment. Independent assortment produces recombinants. This 9 is to 3 is to 3 is to 1. In this 3 is to 3 is for recombinants. Now, let's see this example. We have parents, capital A, capital B, small b, and small a, small b, small b. The gametes will be capital A, capital B from this parent, small a, small b. In F1 progeny, it will be capital A, capital B, small b. When we crossed between F1 progenies, in the F2 progeny, the gametes will be capital A, capital B, capital A, small a, capital B, small b. Now, I'm not completing this boxes because in case of law of independent assortment videos, we already learned how to do the combination and get the phenotypic ratio. This 9 is to 3 is to 3 is to 1 is a phenotypic ratio. So how to get to this phenotypic ratio? By combining different gametes, we already learned. But we know capital A, capital B is the original combination and small b is the original combination. So what are new combinations? Capital A small b and small a capital B. These are new combinations. So in 9 is to 3 is to 3 is to 1, again I am saying this 9 when both the characters dominant, this one when both the characters recessive. So these are what? This 9, both the characters dominant. This one, both the characters recessive. So they will display the parental phenotype. What about these middle two? One character dominant and one character recessive. So these are hybrids. So because of law of independent assortment, in F2 generation, we get 50% recombinance and 50% parental. Now, what do we mean by chromosomal theory of inheritance, which is given by Walter Sutton in 1903? According to the chromosomal theory of heredity, what we can say about law of segregation? The alleles are located on the same locus on homologous chromosome that gets separated during gamete formation. So this is one chromosome, this is one chromosome. This is one chromosome from mother side, one chromosome from father side. They both constitute the homologous chromosome. Now here it is present capital T, here it is present small t. So these alleles are located on the same locus on homologous chromosome and they get separated during gamete formation because in gamete formation one chromosome will be like this in one cell, another chromosome in the another cell. So this is the chromosomal theory for law of segregation. What about law of independent assortment? The pair of homologous chromosome assort independently from other pair of homologous chromosome. So this is one pair of homologous chromosome, 1 and 1. This is another pair of homologous chromosome, 2 and 2. So this will assort independently compared to this. So this is about law of independent assortment. Now, in 1905, one experiment was conducted by William Bateson, Rebecca, and Punnett. What is this experiment all about? So they crossed between parents with capital P, capital P, capital L, P for purple, L for long, crossed with small p, small l, small l. Here, small p for red and small l for round. So capital P and capital L, that is purple and long, dominant over red and round. When crossed, F1 generation, capital P, capital L, small l. For F2 generation, when progenies of F1 generation will cross, what we should get? 9 is to 3 is to 3 is to 1, according to independent assortment. But when they crossed and when they count the number of progenies giving phenotypes, they found that 284 plants are there for the dominant phenotype, 55 plants showing recessive phenotype, and 21 plants each showing the hybrid phenotype. But what is our ratio? 9 is to 3 is to 3 is to 1. Now how many plants? For dominant 284, for hybrids 21, and for recessive 55. Do you think that this ratio is 9 is to 3 is to 3 is to 1? No, it is not. It is not 9 is to 3 is to 3 is to 1. Why it happened? Why they didn't get the result according to law of independent assortment? From here starts the discovery of linked genes. That linked genes segregate together while crossing over produces recombination between them. So what the line is saying is linked genes segregate together while crossing over produces recombination. So this 3 is to 3, the hybrid phenotypes, why they are hybrid? Because of crossing over, because of recombination. But the linked genes do not recombine. Now how we represent the linkage? With the horizontal lines. The horizontal lines are the representation of homologous chromosome. So this is one homologous chromosome, this is another homologous chromosome, and that's why it is written A, B, A, B. So this is one locus having alleles of the gene A. This is second locus having alleles of gene B. Correct? We cannot write like this because this is the same locus on homologous chromosome. So in the same locus, the alleles of a particular gene should only be present. So we can never write like this, that is B below A. We always have to write A below A, either like this or like this. So it is showing the alleles of the same gene and the position is showing the locus on the homologous chromosome. So this is parent, capital A, capital A, capital B, small a, small b, small b. When crossed in the F1 progeny, capital A, small a, as you can see like this, and capital B, small b. Or sometimes, instead of double lines, even with one line, we can denote both the homologous chromosome by writing alleles on the opposite sides. So like this can also be written. Or we can also write with diagonal lines. Now, complete linkage compared to independent assortment. Now, first, let's look into the independent assortment because we already studied about this. This is what capital M, capital D, small m, small d. In the F1 generation, capital M, capital D, small d. Cross with small m, small d. What we call this test cross. What is the ratio of the test cross in case of independent assortment? 1:1:1:1:1:1. From this small m, small d, the gamete is small d, only one kind of gamete. But with small m, small d, we can get four different kinds of gamete. With both the alleles dominant, with one allele dominant, with another allele dominant, with both the alleles recessive. So when we cross, we'll get the ratio of 1:1:1:1. This we already learned in the video of test cross. So this is our independent assortment. Now what if the genes are not separate and recombination cannot happen between them, or independent assortment cannot happen between them? How those genes will behave, or how the linked genes will behave? Now again the same parent with capital M, capital D, small m, small d. F1 generation will be same, capital M, capital D, small d. Now crossed with recessive parent, that is a test cross. Now from this we'll get only one kind of gamete, capital small m and small d. Now from this, what is our condition here? That the genes are linked. So capital M is linked with D and small m is linked with small d. So when capital M is linked with capital D and they cannot separate, so we cannot get capital M and small d, and we cannot also get small m and capital D, because capital M with capital D and small m with small d are very tightly linked. So no recombinations able to happen between them. Without the recombination, we cannot get the recombinants or the hybrids. So now we have only two type of combination. One, all the characters recessive, and second, all the character dominant with one allele dominant and another allele recessive, because this is the combination. So capital M, capital D, small d, and the ratio is 1:1. So instead of getting 1:1:1:1, in case of linked gene, we are only getting 1:1, and both the characters are from parental phenotype. This will show the parental recessive phenotype. This will show the parental dominant phenotype. So we won't get the mixed phenotype. That means one character dominant, one character recessive, and one character recessive, the one character dominant. We won't get these combinations. So what we are getting? All non-recombinant progenies. So when there is complete linkage, we will get all non-recombinant progenies. Whereas in case of independent assortment, we get two progenies non-recombinant and two set of progenies recombinant. So we will get 50% recombinant and 50% non-recombinant in case of independent assortment. Whereas in case of complete linkage, it is always non-recombinants. Now, next is crossing over with linked genes. Now, the genes are linked, but it is not always the case that genes are very tightly linked that they cannot recombine ever. So some crossing over happen between the genes, and such genes are called incompletely linked genes. So you can see it here. When there is no crossing over means all non-recombinants. When there is crossing over, so we will get recombinants, 50% recombinant and 50% non-recombinant. So even in the linked genes, some crossing overs are allowed. So for closely linked genes, crossing over does not take place in every meiosis. In every meiosis, there won't be crossing over like a normal genes, but some crossing overs in some meiosis is allowed. If crossing over occur in any meiosis, half will be recombinant, half will be non-recombinant. If crossing over does not occur, all recombinants. The frequency of recombinant gametes is always half the frequency of crossing over. And the maximum proportion of recombinant gamete is 50%. Recombination cannot exceed more than 50%. The 50% is the maximum proportion of recombination. Now, a test cross between a homozygote and a heterozygote will produce half heterozygote, half homozygote. We already saw this in the last slide. Each crossover takes place between only 2 of the 4 chromatids of homologous pair. As you can see here, these are 4. 2 homologous pair means 4 chromatids, right? Now the crossing over happened between this middle 2. So out of 4, only in 2 chromatids the crossing over happened. And the crossover takes place in about 50% of meiosis. So what to remember from here? That when no crossing over, all non-recombinant. When crossing over, 50% recombinant, 50% non-recombinant, and the recombination proportion cannot be more than 50%. Now, this is a crossing over between linked genes. So this parent is crossed with this parent. From this parent, we will get only one kind of gamete. From this parent, if no crossing over, then we will get only the parental gametes. If crossing over happens in some meiosis, in that case, we will get parental gametes also and the recombinant gametes also. Correct? So what we know is linked genes always produce non-recombinants. But in linked genes also, some crossing over is allowed in some meiosis. So when crossing over happened, they will form 50% recombinant and 50% non-recombinant. Clear? So from linked genes, we are getting only non-recombinants when no crossing over. When some crossing over happen also, in that case, we will get 50% recombinant and 50% non-recombinant. So if we see the proportion, here 50% is non-recombinant, here 100% non-recombinant. Correct? So the proportion of non-recombinant is always more than the proportion of recombinant. So even in linked genes, some crossing overs are allowed. So the meiosis where crossing overs does not happen, those meiosis will produce all non-recombinants. The meiosis where crossing over happened, there it will produce 50% recombinant and 50% non-recombinant. So in total, if we see here 50% non-recombinant, here 100% non-recombinant. That means the proportion of non-recombinant will always more than the proportion of recombinants. And this what we can get, we can see here. So when linked genes are test crossed, in that condition, in the next generation, we are getting 53 plants each with parental phenotype, and we'll get 8 and 7 plants with recombined phenotype. So as you can see, the number of non-recombinants are more compared to the number of recombinants. Now remember this numbers 53 and 53 for non-recombinants, 8 and 7 for recombinants. Why? For the calculation of recombination frequency. What do we mean by recombination frequency? The percentage of recombinant progeny produced in a cross. That is called recombination frequency. The formula is number of recombinant progeny divided by total number of progeny into 100. I asked you to remember the numbers 53 for non-recombinants, 8 and 7 for recombinants. So number of recombinant progeny 8 plus 7. Total number of progeny 53 plus 8 plus 7 into 100. That is 12.2%. So the recombination frequency for this example is 12.2%. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.